The human brain comprises only 2% of our body mass, yet accounts for 25% of our daily metabolic demand. It is the most energy-intensive organ we have. This high metabolic rate creates a constant stream of byproducts. Specifically, neural activity generates metabolic waste solutes, including a protein called amyloid-beta. In most organs, these byproducts are managed by the lymphatic system, a network of vessels that collects interstitial waste and returns it to the general circulation. Looking at this cross-section of the blood-brain barrier, we see a highly regulated membrane that separates the brain from the blood. This barrier is so effective that the brain lacks any traditional lymphatic vessels to drain waste. Without a drainage system, these metabolic proteins would accumulate in the spaces between neurons, eventually interfering with synaptic function and contributing to neurodegenerative disease. This creates a biological mystery. How does an organ with such a high metabolic load maintain a clean environment without traditional plumbing? The answer was discovered in 2012, a specialized waste clearance network known as the Glymphatic system. This system doesn't rely on new vessels. Instead, it uses a perivascular network that piggybacks on the brain's existing arteries and veins. It is a specialized infrastructure designed to utilize the space around blood vessels to flush the brain parenchyma. This diagram illustrates the process. Cerebrospinal fluid enters along the arteries. It passes through AQP4 water channels on astrocyte end feet into the interstitial space. This convective flow picks up metabolic waste surrounding neurons and finally exits along the veins to the systemic circulation. This dynamic mechanical process maintains a continuous exchange of fluids within the skull, effectively washing the entire brain parenchyma. This plumbing system is state-dependent. It remains largely disengaged while we are awake. The system requires the specific electrical and chemical environment of deep, slow-wave sleep to function efficiently. During the sleep phase, the space between brain cells increases by 60%, significantly reducing resistance. Simultaneously, the rhythmic pulsations of slow-wave sleep act as a pump, driving cerebrospinal fluid through these expanded pathways to clear out accumulated solutes. Sleep provides the mechanical conditions necessary for the brain to clear the metabolic byproducts that build up during wakefulness. As we see in the young panel of this diagram, a healthy brain maintains an efficient, unobstructed flow of fluid across the tissue. However, as shown in the old panel, aging can impair this process. The AQP4 channels often lose their polarization, moving away from the blood vessels and slowing the rate of fluid exchange. In the Alzheimer's disease panel, we see a feedback loop. As waste clearance slows, proteins like amyloid-beta accumulate, which can physically obstruct the pathways and further reduce the efficiency of the flush. This suggests that the gradual decline of glymphatic efficiency is a significant factor in the progression of age-related cognitive disorders. While age is a factor, experimental evidence suggests we can influence the efficiency of our brain's plumbing through lifestyle choices. Research on rodents indicates that body posture matters. Sleeping in a lateral or side-lying position appears to clear solutes more effectively than sleeping on the back or stomach. Dietary choices may also play a role. Intermittent fasting can increase the polarization of AQP4 channels, potentially improving the rate of fluid exchange. Ultimately, these interventions depend on prioritizing deep sleep, which provides the physical force necessary to drive the entire clearance process. By understanding these mechanical pathways, we can make choices that support the brain's natural waste removal system throughout our lives.